Okay, so we had quite a lively discussion, I think. I think that's fair enough to say. And so what we were really looking at was, what UK resources there are currently available to facilitate research into human knockouts, and really what the gaps are and where we see, we would like to sort of have, what we'd like to have plugging those gaps. Our discussions were sort of quite broad-ranging, a little bit divergent here and there, and quite heated. So we've definitely had some good viewpoints put forward. And we really see the process as sort of being a stepwise process. It made sort of intuitive sense for us that sort of there's a discovery phase, which is sort of discovering the variants, the total pool of variants, a validation phase. We subdivide it into these three areas. A validation phase where we look at actually, what is a knockout and how do we know it's a knockout, and then sort of that clinical impact, that sort of phenotype phase. And so we sort of segmented our discussion into those three areas. Coming back to the discovery phase, I mean, a lot of the data is already available. There's initiatives underway to get 1 to 5 million more people sequenced, and so it's not simply a case of we need more data and more people. We thought maybe adding some sort of targeted element to that data collection, maybe towards populations which, draw out the most information. So the consanguineous populations, maybe a diversity across those. We talked about, if we could have some sort of way of targeting those populations within the UK, and maybe even sort of reaching outside of UK with putting, say, maybe grants or something forward, or encouraging, for example, collection of individuals' genomes from, say, the Pakistani population or other known populations, such as those. But also hand in hand with that is the metadata that we collect with those individuals as well, and sort of reassessing that and thinking, maybe we would want enhanced validation at that first, enhanced metadata at that first point of contact, and also bringing some sort of standardized nature to that, consistent, something akin to what's worked very well for the UK Biobank there as well. We talked about the types of data that we would want in a perfect world and also in reality. I mean, we've got whole genome sequencing data. We talked about, we don't actually look at all of the whole genome sequencing data, so we questioned, is that really what we want? Would we prefer just an exome data, for example, and some sort of SNP array? Would that be more informative?
Plus RNA-seq.
One of the advantages of going down that route was it saves money to allow us to get RNA-seq. And so maybe in the perfect world would have whole genome sequencing data and RNA-seq. There could be some sort of alternate approach to that. We also touched on no tissue type fits all, and so what might be relevant in the blood, especially with respect to RNA-seq, may not be relevant to all of the mutations or variants that we find and across individuals as well. Sort of moving on into, so that sort of segues into the validation segment, and we sort of really split validation into sort of two different areas. One is validation that this knockout is really a human knockout, so it's functional at the gene level, and the other is at the organism level, so the phenotype and the clinical impact. We asked sort of rhetorical questions, but also philosophical questions about what is a knockout. It makes sense that a knockout is a frame shift, but we heard different examples of when a frame shift actually doesn't lead to a reduction in the RNA or the protein itself, or just leads to a slightly reduced number, and maybe there needs to be some sort of, it's much more nuanced than we like to conflate it down into just knockout, but really maybe we need to look more at what we're actually calling a knockout and be a bit more explicit with how we describe a knockout. We also talked about making that data accessible. So as you go from that sort of validation to that phenotyping aspect, that's often where a wet to dry lab or dry to wet lab interface exists. And if we've learned anything from history, that those interfaces are quite difficult to make smooth. And so we thought about what we would really need there to enable essentially the data, this complex data, really valuable data, to be really properly exploited would be some sort of focus on making it accessible to, you know, the basic humble cell biologists such as myself. And we also talked about how do we then inform the clinic? I mean, what do we need to do next to really give information to the clinic? The hope is that a patient will come into the hospital and get sequenced, and then a week later the clinician will be able to give some sort of diagnosis. That might work for low-hanging fruit, but in reality we're going to have to go down some sort of bespoke route where we explore these knockouts in various models. And we talked about what those models should be. Is the mouse modeling era over? Is it dead, you know? Or has there still got some legs in it yet? And so that we had some heated debate around there, and maybe there's a middle ground. We can use better cellular models and some of the newer technologies such as CRISPR to really perturb these genes and sort of really try to understand what's going on in a sort of more quickly accessible format to sort of feedback. We also talked, sort of just finishing up now, about how to get around very small sample size when you only have one patient with a particular mutation, or maybe two. I mean, how do we deal with that? Does that always have to happen in that patient's tissue? And how do we account for all the other mutations that are interplaying within that patient's tissue? And also, how do we incentivize recall once we've found these patients that we want to bring in their family members and what have you? And that sort of went on to the ethics sort of area of consenting and what have you. So I think that's where we sort of left it there.
Thank you very much.
Okay.
Hi everyone. So we were looking at the industry perspective on research into human knockouts, distilling the discussions. We focused on a few different areas, including the roles of human knockout for industry, considerations of ideal knockout cohorts from an industry perspective, the role of industry in an online catalogue of human knockouts, and whether and why industry should work in the NHS in the UK, and the perceived benefits to working here for the industry, NHS and UKPLC. So in terms of the first one, perhaps looking at the benefits of human knockouts, examples were given about where it's been very useful in drug discovery, such as with PCSK9 inhibitors, and some key characteristics were brought to the surface, such as circulating proteins, gain of function, that these were rare diseases also relevant to common polygenic disease. However, those key characteristics, whilst ideal for drug development, may not be the common model and may not apply, and frequently won't apply to the majority of drug development. Therefore, human knockouts can still have potential benefits in that space, for example, helping us to undergo target validation and understand potential safety and efficacy of perturbing the target before great investment in phase 3 trials has happened. However, it was pointed out that there still needs an awful lot of upfront investment and research to understand more about the targets than perhaps was done historically. Furthermore, as pointed out in the earlier talk, just because something is shown up as a Mendelian disease doesn't mean that perhaps you can't modulate other targets in order to overcome that. So it's seen in human knockouts. It's not the be-all and end-all. It's another tool to help understand and help in drug development, and perhaps might have utility in prioritising targets to take forward in a portfolio. No, sorry, it's a fair bit of glare. So that was sort of the first. I think ultimately it was felt industry has enthusiasm in this space, but it's looking really for projects and ways to materialise it, and it's not quite there yet at the moment. Therefore, what cohorts would be ideal from an industry perspective? And lots of discussion was on this, and there were two different complementary approaches. The first was a very specific approach where phenotypes of interest to a particular industry are focused on, and an industry may be more involved in helping to perhaps fund these studies, and in turn they would need some form of perhaps short exclusivity in return to help them perhaps until phase 2A, 2B benefit from their investment in terms of identifying new targets through knockout studies. Then there's the more general, earlier exploratory discovery approach, which would take a community of practice consortia, such as IMI, and the cohorts of interest might include more rare disease cohorts, patients presenting younger with common disorders. It was wondered whether phenotyping could be done, for example, in the urine or plasma to identify interesting cohorts to take further, particularly because urine phenotyping is quite cheap compared to, as pointed out, say, phenotyping Parkinson's disease, where you'd need MRIs for each patient. And so those different approaches might be used to help enrich a population which may have human knockouts. It was also, there was some discussion of where would be the optimal places. This should be done in primary care or secondary care, and there are benefits and disadvantages of both. There was a little bit of discussion as well about intellectual property. So if I understand correctly, often that's applied at the point where the chemistry, where you've got a lead molecule. But it might be harder to protect novel findings from bioinformatics analyses, where you're looking at perhaps new diagnostic tests, and thought would need to go into that. Ultimately, industry may be more of an observer in the earlier discovery stages and more hands-on with the recruiting or funding studies that will take patients with their phenotype of interest. The next part was about an online catalogue of human knockouts, bringing together, for example, data from UKPGx, 100,000 Genomes Project, possibly NOMAD. I think there was interest in this. However, from an industry point of view, the most interesting thing wouldn't be just using it as a data resource, which is publicly available, but actually, could there be a recall mechanism? So patients flagged up of having potential interesting knockouts, could they be recalled in order to undergo deep phenotyping to understand more about the systems which in pharma, that particular company is interested in understanding? Such an approach, though, would need a trusted intermediate, such as NHS as a physician, to go between industry and that patient and to ensure that all processes are duly followed and to protect the patient's rights in overseeing that. And lastly, so what are the advantages for industry in working in the NHS? Well, it was pointed out that it's an opening, welcoming research environment, and the NHS is unique. It offers longitudinal follow-up, particularly strong in the area of primary care clinical data, and now there's NHS unique identifiers that are going to allow continuous longitudinal follow-up. What's the value to the UK? Well, it was, having industry doing research here. Well, patient benefit. The launch market is almost the same as the space in which the initial trials are done. So if we're identifying UK patients carrying out research here, hopefully it will be those cohorts that also benefit from drugs down the road. And also really helping to stimulate the NHS in terms of digital transformation, giving them an incentive to transform, an incentive to help bring more genomic medicine into the NHS, and that will help move towards a more preventative model which will help patients, but also open up new potential therapeutic areas which will be of interest to pharma. I think I may have missed things, which I apologise, but I think those are the main points in our discussion. Thank you.
So thanks to Anna, who was chairing and writing down quite a lot of this stuff. So we first tried to identify all of the issues that we thought may need to be targeted, and then we tried to identify solutions to each one of those issues. So the first one that was brought up this morning was the social responsibility in drug development and how can that be addressed. But we also thought that there was a linked issue in terms of the social attitudes to research and how that varies in different societies and different groups of people. As has been mentioned before, it is very important how do we organize the recall for phenotypes. And we then went on, because there are differences in the recall if you are totally interested in the results because you have a disease or because you haven't, whether you're a control or not. And how that communication is done and what is what needs to be said, etc., etc. Then we thought about how do we do this targeting of consanguineous populations, because it may have negative impact into these populations. They may get to have labels of consanguinity, and then it may impact on their social norms or on their social acceptation of things that may well be different, and how we need to think about how to address those. We had a little bit of discussion on the funding, the fact that we then agreed to fund genetic studies for smaller groups. That means the funding is not infinite, so it means that there is funding that is not going elsewhere, and we had a bit of that. And we did have a discussion about the media portrayal of knockouts, mutants, human experiments, and whether that could be sort of dehumanizing or not for the people that are involved in these trials. What is the next scandalous media headline that you may find around this issue? So in terms of the social responsibility, we needed to make sure that in the discovery process, different populations need to be targeted, the women, the young, and the old, not only young male that are white, minorities, etc., etc. So your clinical address have to target different populations. The populations that are used in discovery have to be benefited some way in what is it. So that Finnish model where if you have a drug that has been using those drugs, then should be cheaper for the Finnish population in general or not, may be easy to be discussed or not. And we didn't know very well how to organize this question of when pharma companies have been benefiting from a certain population or a certain group of deceased individuals, how is that given back? We had a small discussion about whether the IP should be shared, but that brings a lot of more complication. So we thought of just encouraging schemes of societal responsibilities. But we thought that those should be encouraged, talked about name and shame companies. And so if I go more or less in order, in terms of targeting consanguineous population, I'm not going to go in order. We thought that we needed to be careful about how to handle the social stigma and how the same populations feel their social stigma. So for us, it may be, or for white population, may be more relevant to be considered that you're marrying your cousins, whereas for them, for other populations, it may be that you have a mental disease or you end up having something that is different. The findings of that should be discussed with the communities. I think the best, we have to solutions there. One was engaging the communities early, so they have to be engaged with the research group and the ethics committee. And then maybe, a way to pay back would be offering testing, genetic testing and genetic counseling to those communities that are that have been specifically useful to discover some of that. In terms of the recalling, we had a long discussion on how to recall and what to do when you recall individuals. So we thought that the initial consent had to be rather clear in terms of how, in what situation are you going to ask them back. And then there has to be another consent that has to be agreed with the ethics committees on what is really said in the second consent and why do you approach. We thought that there needed to be somehow, and we didn't know how to organize that, a limit on how many researchers can contact particular populations, especially, people with sexy knockouts. How many times? How do we ensure that there is a limit to the request to those populations? Could we centralize the ethics committee? Could we use one ethics committee, or could we use that information? We thought that it was very important to keep in the database what information was given to the participants when. So, because also that information may change in the near future in relevance. So, things that are not medically relevant today or thought not to have a deleterious effect in health, it may come later on visible that they might be. And therefore, we then went on how to consider or how to engage in a dynamic consent, because the consent will change because new information is highly likely to come on board. There are other situations where new information is not. Here, for sure, new information is going to come. So that's about the recall. With the social attitudes to research, the difference between different countries, we thought of the difference between the NHS. UK has a certain values because they are related to having a service like the NHS, which is universal, versus the USA, who have many different values because their medical care goes under a very different payment pathway. There are cultural, so the culture may be different, social classes may interpret this different, education levels have to be taken into account, and age. There are people who are more likely to say yes to everything, and there are people that are less likely to say that. And we wanted to make sure that different peoples end up not being excluded from research. Ethnic minorities, disconsanguineous population, non-English speakers, people with low IQ. And we said that, as before, the implications are very different for the people that are identified as knockouts, whether they have health implications for themselves or not. And one of the things that we said, I don't know where we say this, in the media thing, how to address the people that are knockouts. And we thought maybe knockouts is not a very good term. And you see, so whether we should, one of the recommendations should be to use a less media attentive term, like homozygous loss of function, or people that have loss of functions, or things like that. But knockouts didn't seem to be a very proper term, which has been used in all of the communication. Did I leave anything else? And we need an important education to all, not only the participants, but also the society, on what is the meaning or importance of this particular loss of function, so that we can all then engage and it can be beneficial for society in general. And I think that's it.
Thank you very much.